So I mean, maybe we're jumping ahead, but why not get just totally different programs and different circuits that end up with the same kind of segmented phenotypes?
Well, because, as any zoologist from the last 200 years will tell you, segmentation is too complex to have arisen more than once. Too complicated.
No way.
I know I'm sounding a little bit disrespectful, but that is essentially what the argument boils down to. Look how complex segmentation is. You duplicate the muscles, you duplicate the nerves, you duplicate the endoderm, you duplicate the ectoderm. Everything has to be connected and work, and it's the same over and over from the inside to the outside. Too complex.
Huh. How many times is it thought to have evolved independently? I mean, the more modern incarnation.
Well, you got sort of at least 3 hard to argue with times. Arthropods, annelids, and vertebrates. Now, let's just go backwards. You got the vertebrates. Who are their close relatives? Sea squirts, starfish. Not a lot of segments happening.
Right.
Okay. Then you say, okay, the arthropods, who are their close relatives? Other things with cuticles. You have this group of animals with cuticles that there's still lack of clarity. Are they segmented? Are they not? And things like kinnorynx, preapulids. Kinnorynx, better example, loricifera. Are they segmented? Are they not? Then we get into the whole corner of what's a segment, which is verging on philosophy.
Yeah, sure.
And so that's the discussion you have to have when you start to talk about those guys. And then annelids, who are their close relatives? Mollusks, maybe flatworms. The close relatives of annelids, and when I say close relatives, I just mean sort of within the spiralia, the internal relationships of those organisms are still very poorly understood. So that's one problem. And then, the embryology of many of those guys also still poorly understood.
Right.
So, but those 3 cases, pretty inarguable. You look at them, you say, well, those are segments by anybody's definition.
Right. And that's still such an incredibly broad level of organization. I mean, within any of those taxa, is there an argument that there is are people pushing that there are finer levels of splits or yet more splits? I mean, what's the upper maximum on the number of evolutions of segmentation that people are willing to accept?
Oh, that's an interesting question. I couldn't tell you what an upper maximum is that I've seen, but 6 or 7 times maybe, depending on what you think is segmented.
Right.
7
Back
or
to
8
philosophy.
times. Right, because people want to talk about annulation versus segmentation. Like, what if I see grooves on the outside, but then I cut it open and I don't see subdivisions on the inside?
Right.
Does that count? How far inside do I have to go and be subdivided to still count?
Yeah, those time scales, it gets pretty tricky on that front. So can we...
it does.
This is not, Cassandra, this is not even really what we intended to talk about, but this is fun. So indulge me one more thing. You said a minute ago that Sydney Manton did a lot of work that, the species that she studied haven't been studied since. What's the reason? Why?
It's difficult to access. So she did a lot of work on some South African and I think Caribbean, I have to go back and just check on that, but a lot of tropical and subtropical onychophorans, for example, multiple species of onychophorans. Onychophorans are a pain to work with. You have to go tramp through forests, dig them out of rotting logs. I am not sure that anyone's really been able to sustain sort of over many years a sustainable colony in a lab. And so it's just difficult to get the specimens. Yeah, I think that's one of the main reasons. Okay. They're not easy to keep in a lab, so you have to go get them. Yeah, I went and collected some in Australia.
I'm so jealous.
It was really an incredible experience. I was going to Australia for a meeting, and I was, reading Sydney Manton's work and trying to learn about onychophorans and just invertebrate zoology in general. And I thought, I wonder, one of the experts who has worked on development and life history is this guy, Noel Tate, who's in Sydney, and maybe I wonder if he would see me. And I called him and I said, could I come and learn about, just, chat about onychophorans? He was like, yeah. So I showed up expecting that we would sort of chat in his office. Maybe he would show me some specimens. He was basically ready to go hiking for a day to take me to the Blue Mountains to find onychophorans in the wild.
That's pretty fantastic.
Him and his wife.
And you did.
It was amazing. His wife is a geologist. So we're driving through, and she's like, you see these formations here? They show you that this is of this and this age. I don't even know any geology, so I couldn't tell you what. But it was incredible. It was, we went to this eucalyptus forest. We tramped through. You find the rotting eucalyptus logs. The kangaroos are hopping by on the side of the road. You hack open these logs. You find the onychopherans. I'm not even kidding. We stopped and barbecued kangaroo meat on the way back for lunch.
Perfect.
It was amazing.
Like an epic day.
It was epic. And Noel Tate is a legend, and I just have to thank him for introducing me to onychopherans like that. It was really incredible.
Well, that's so great. You've seen them. I've talked about them in many classes over many years, but they always seem like some magical group that, you know, there's literature about them, there's films of them, but I will never see one.
Right.
And
They're real. Even I am not a field biologist. Even I could find them. Once I was shown how to look for them, I could find them. Yeah.